This presentation is about the metabolic process of glycolysis in the human body. The focus will be on the main purpose of each step of glycolysis. At the end of the glycolysis summary, we'll look closer at the regulatory steps of glycolysis. In the research paper selected for this project, a regulatory step of glycolysis is blocked in cancer cells in mice. The effects this has on tumor proliferation was recorded and analyzed. The research paper investigated the effects of inhibiting a glycolytic activator, PFK-FB3, in tumor cell epithelium. Cancer cells, especially their outer cells, are hyperglycolytic. This means that they depend on and demonstrate increased glycolysis to fuel their growth.
Glycolysis is the conversion of one molecule of glucose into two molecules of pyruvate, two NADH molecules, and two net ATP. In addition to providing important metabolites, glycolysis is vital to the human body because certain tissues, such as the brain and red blood cells, rely solely on glycolysis for energy. Glycolysis can be broken down into two phases, the investment phase and payoff phase. The investment phase requires the use of two ATP molecules and produces two molecules, only one of which will continue through the rest of glycolysis, and that is glyceraldehyde 3-phosphate, or GAP. Step 1 of glycolysis is the conversion of D-glucose into glucose 6-phosphate, and this step is catalyzed by hexokinase. This prevents the glucose molecule from leaving the cell and begins the process of turning it into a high-energy molecule. Hexokinase uses a divalent ion as a cofactor like magnesium. We have invested 1 ATP at this point. Step 2 is the rearrangement of glucose 6-phosphate into fructose 6-phosphate. This step is catalyzed by phosphoglucose isomerase and involves the opening of a 6-membered ring and closing it into a 5-membered ring. Carbon 1 is now outside of the ring. Step 3, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, is a key step in the regulation of glycolysis. This step adds another phosphate group to carbon 1 of fructose and is regulated by phosphofructokinase, PFK. The purpose of this reaction is to trap the sugar in its more unstable ketose form because, unlike glucose, fructose can be readily cleaved into 2 3-carbon fragments, one of which is GAP. Step 4 is the splitting of fructose 1,6-bisphosphate into 2 3-carbon sugars, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, GAP. This step uses the enzyme aldolase. Step 5 uses a vital enzyme involved in the regulation of glucose, and that is triose phosphate isomerase, or TPI. This enzyme interconverts DHAP and GAP, the 3-carbon products from the cleavage of fructose 1,6-bisphosphate, between one another. This prevents the waste of GAP, an essential resource in the formation of pyruvate, because DHAP is converted into GAP only so fast as GAP is used up. GAP is the only molecule that continues into the payoff phase of glycolysis. The importance of this enzyme is demonstrated when someone has TPI deficiency, the only glycolytic enzymatic disease that is lethal.
Now here we are at the payoff phase with some glyceraldehyde 3-phosphate, or GAP. For every 1 molecule of glucose, we eventually end up with 2 molecules of GAP to feed into the payoff phase. So for the rest of glycolysis, the products should be multiplied by 2 to get the final count of the products. In step 6 of glycolysis, glyceraldehyde dehydrogenase adds an inorganic phosphate to GAP, producing 1,3-bisphosphoglycerate. 2 important discrete events happen during this step. 1. GAP is oxidized by NAD+, or the NAD+ is reduced by GAP into NADH. Then a phosphate group attacks the GAP molecule, which releases it from the enzyme and releases an additional hydrogen atom. This happens twice, so we have a gain of 2 NADH and hydrogen atoms. Step 7 is similar to step 1 in glycolysis, in that the enzyme used, phosphoglycerate kinase, also uses a divalent ion like magnesium as a cofactor. This helps orient the negative phosphate groups into the right positions on the molecules. This step transfers a phosphate group from carbon 1 to ADP to form ATP and 3-phosphoglycerate. Because this is happening for 2 GAP molecules, we have broken even in terms of ATP investment and payoff at this point. We have a net of 0 ATP. Step 8 uses the enzyme phosphoglycerate mutase, which relocates the phosphate group from the 3rd carbon to the 2nd carbon, forming 2-phosphoglycerate. This is accomplished by the addition of a phosphate group to the second carbon and removal of a phosphate group from the third carbon. Step 9 uses the enzyme inhalase, which removes a molecule of water or dehydrates 2-phosphoglycerate and forms phosphoenolpyruvic acid or PEP. Step 10, the final step of glycolysis, uses pyruvate kinase. This enzyme transfers a phosphate from PEP to ADP to form another ATP molecule and pyruvic acid. Since this happens twice, we have a gain of 2 pyruvic acids and 2 ATP molecules. To summarize, the net products of glycolysis are 2 pyruvates, or pyruvic acids, 2 NADH, 2 ATP, and 2 hydrogen atoms. In metabolic pathways, enzymes that catalyze irreversible reactions are usually sites of control. In glycolysis, the reactions catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase are irreversible, and so are the regulatory steps of glycolysis. The control of glycolysis is a little different depending on the tissues where it takes place. The regulation of glycolysis presented here will be focused on skeletal muscle tissue, but some notes about the regulation of glycolysis in the liver will be mentioned. Hexokinase is the primary regulatory step in glycolysis. High concentrations of its product, glucose-6-phosphate, inhibits the activity of this enzyme because its product is not being used up, which signals to the cell that glucose no longer needs to be broken down. Phosphofructokinase, PFK, is the most important control site in the glycolytic pathway in mammals. High levels of ATP allosterically inhibit the enzyme. A high concentration of ATP means that we are not using up our ATP. We do not need to make more. ADP and AMP competes with ATP for the regulatory site. As we use up ATP, the concentration of ADP and AMP increases and will begin to outcompete ATP for the regulatory site. Why is phosphofructokinase instead of hexokinase the main regulatory step in glycolysis? That is because glucose-6-phosphate is not only used in glycolysis. In skeletal muscle, glucose-6-phosphate can be converted into glycogen for energy storage. Phosphofructokinase is the committed step, step 3 of glycolysis, because it is unique to glycolysis. Also, the inhibition of PFK can indirectly inhibit hexokinase. As the substrate for PFK, fructose-6-phosphate, builds up, some of it is converted back to glucose-6-phosphate because it's in equilibrium with glucose-6-phosphate. Pyruvate kinase is the third irreversible step in glycolysis. This enzyme controls the efflux or output of products of glycolysis, pyruvate and ATP. High levels of ATP will allosterically inhibit pyruvate kinase. The product of PFK, fructose-1,6-bisphosphate, activates pyruvate kinase, promoting its activity to deal with the incoming substrates for the enzyme.
This experiment tested a method of combating tumor cell metastasis by normalizing tumor cells by inducing PFKFB3 deficiency. PFKFB3 is a glycolytic activator in endothelial cells in tumors, and endothelial cells demonstrate hyperglycolytic activity, so they are vulnerable to glycolytic inhibition. Normalizing tumor cells actually improves their currently existing vessels, making them healthier in a sense. Tumor vessels are typically structurally and functionally abnormal, which impairs perfusion of oxygen and nutrients to the tumor. In response, tumor cells escape from this hostile environment via invasion, intravasion, invasion to the circulatory or lymphatic system, and metastasis. Here is an image from the National Cancer Institute depicting tumor metastasis. Normalized vessels improve perfusion to the tumor, reducing stress on the tumor, and reduces VE cadherin endocytosis. VE cadherin endocytosis is the ability of the tumor cell to squeeze past blood vessels to travel to other parts of the body. As you can see on the left, a cancer cell can usually move between the endothelial cells of vessels, which can lead to infiltrating other parts of the body, including the lymphatic system. The endothelial cells after glycolysis has been inhibited are more tightly bound together. Pericytes, the cells in green, help maintain the integrity of vessel walls and when activated promote angiogenesis, or the formation of new blood vessels. This requires a lot of energy. Inhibiting glycolysis makes pericytes less active. The normalization of tumor vessels is an emerging approach to cancer treatment that, besides reducing tumor invasion and metastasis, improves chemotherapy drug delivery and effectiveness. Improved perfusion to the tumor means that more drug is delivered to the tumor, and many chemotherapeutic drugs require oxygen as a part of activating their therapeutic effect, another nutrient that is more readily available after the normalization of tumor vessels. The results for this experiment were collected by recording tumor weight, cancer cell proliferation, cancer cell invasion. PFK-FB3 inhibition improved chemotherapy, reduced the growth of primary and metastatic tumors, and impaired cancer cell dissemination. These findings warrant further consideration of targeting tumor EC glycolysis as an adjunct to traditional cancer treatments.
Glycolysis is a metabolic process that provides immediate energy and fuels a lot of tissues and processes in the human body. Red blood cells and the brain can only get energy from the metabolism of glucose molecules. In the case of the research paper, rapidly growing cells such as in cancers rely tremendously on glycolysis. Inhibiting glycolysis, while it may seem to confer some benefit to the primary tumor by improving its perfusion, diminishes one of the major advantages cancer has, and that is metastasis, as seen on graph O. Thank you for watching our presentation, and I hope this made the intricate process of glycolysis more interesting and easier to understand.